So welcome to the session on reporting on genome editing. My name is Diana Kenny, and I'm a science writer and editor at the Marine Biological Laboratory in Woods Hole, Massachusetts. And our panel today includes science journalists from 5 different nations, whom you will meet in a few minutes. And we're here today to talk about a genetic engineering technology that was introduced in 2012 and has since spread like wildfire throughout the scientific research community. This technology is called CRISPR-Cas9, and it gives scientists the ability to precisely target and manipulate defined stretches of DNA in virtually any species, including human beings. Now, this technology seems to have unlimited applications in many fields, especially now there's a lot of activity in medicine and in agriculture, and it has already spawned $1 billion startup industry. So today we're going to focus especially on the application of CRISPR-Cas9 technology to editing human embryos, which is research that's at early stages in several nations. And when changes are made to the DNA of embryos, those changes are heritable by future generations. So if and when this technology reaches clinical practice, medical science will essentially have begun to control the course of human evolution. So given the far-reaching implications of this technology and the speed at which it's being developed, and the situation that many people in the public seem unaware or confused about what it is, this presents many challenges to science journalists, and those challenges are not the same from nation to nation, because the story is not unfolding in the same way in different nations. So in our panel discussion today, we'll learn about some of the specific cultural, economic, legal, historical, religious factors that are affecting coverage of human genome editing in various nations. And we hope that when we open up the discussion to you at the end of the session, that we can broaden the conversation on commonalities and differences in covering this very fast-moving technology. So I'd like to introduce you to our moderator, Carl Zimmer. Carl has been writing about science for the New York Times since 2004. He writes a weekly column called Matter. Carl is the author of 13 books. He's currently working on his 14th, a book about heredity called She Has Her Mother's Laugh, to be published in 2018. Carl began his career in science journalism as a senior editor at Discover, and he's written hundreds of articles for many other magazines, including Wired, National Geographic, The Atlantic. He is a national correspondent for STAT. He appears regularly on Radio Lab, and among his many awards, he is a 3-time winner of the AAAS Kavli Science Journalism Prize. Carl also teaches writing at Yale University.
Thank you, Diana, and thank you all for coming. When I was asked to give this talk, I definitely jumped at the chance because I personally have felt like CRISPR has kind of taken over my life. Let's see if we can get this. So this is CRISPR. Just to remind you, this is an enzyme that can basically cradle a piece of RNA, and you can design it so that it can go into the nucleus of a cell and be able to actually recognize a specific segment of DNA. The chemical details we know are different than this now, but this gives you the basic idea that you can basically lock in across the genome at one particular spot. You can cut this and make a break. And once you make that break, DNA is going to dissolve away in that area. One thing you can do with this is you can basically snip out a piece of DNA. So if you just want to delete a tiny piece with some accuracy, CRISPR is something that lets you do it. And an alternative is that you can put in a gene that you, or just a segment of DNA you want to replace the old one with, And thanks to the way that DNA repairs itself, it will take in that new piece of DNA, and voila, you've reprogrammed your DNA quite precisely. So, in 2013, CRISPR hits, and a lot of us journalists are like, oh, what's going on? What is this, and what implications will it have? And I personally started writing about it within the first few months, and one of the things is there are all these possibilities that got raised. So, for example, you could use it to reprogram mosquitoes. What I found personally was that that there were a lot of people just want to know, well, what the hell is this? And so I wrote a longer piece for Quanta trying to kind of lay out how CRISPR was discovered, how it, and just how it works biologically. What really astonished me was that there was just this incredible hunger that people had to know what was going on. So I was contacted by the folks at Radiolab who I'd worked with before, and we did a CRISPR episode where really I was just sort of explaining the basics, and they said that this episode was hugely popular, and I think because people were hearing about this and they want to know just the basics of what is it, and it's sort of basically taken over my life, and so my most recent project is working on a book about heredity in general, but CRISPR really had to be a part of it. But it's kind of just echoing what Ed and Steve and Mary were talking about today. You get a little nervous writing about this sort of cutting edge stuff in a book because you kind of know it's going to go out of date pretty fast. Case in point, the technology of CRISPR has moved so fast that I'm literally trying to proofread the book today, and I might try to get this in there. This just came out about actually one more step towards using CRISPR just to adjust individual letters in DNA. And so this field is moving incredibly fast just in the basic biology. But there's a whole extra level of reporting that we do with CRISPR. Is like, well, what could this realistically be used for? So there are basically two main, well, so there's several different ways, but one of the key ones is what you could call kind of gene therapy. So this is a diagram just showing you how you could use CRISPR to reprogram cells that you would then put into a diseased heart. There are other sort of gene therapy type applications that people want to use. But of course, like what fixates everyone is the possibility of using these kinds of technologies to reprogram human embryos. In 2015, researchers in China published a study where they were using CRISPR on embryos at these very early stages, and that really kind of reframed the whole discussion. And so we really are faced with, what are the ethics of even doing any research on human embryos with CRISPR, and should we someday be using this as a sort of therapeutic treatment? Will that lead to enhancement? I mean, there are all sorts of possibilities that it raises. So, for me, one of the exciting things about this session is that we were able to have an international perspective on all these issues, because in each country, it's a different story. And so this is a little map of where everybody is from. And so I am going to turn things over to Antonio Regalado from Technology Review. He will talk about CRISPR in the United States, and then Xiaoshui Chen will from the Intellectual, we'll talk about what's going on in China. Then we will jump over to Australia, where Natasha Mitchell from the Australian Broadcasting Company will talk about things there. Valeria Roman from Buenos Aires will talk about what's going on in Argentina and Latin America more broadly. And then finally, Pal Ghosh from BBC will talk about the issues in England as well as Europe. So I'm going to be a ruthless taskmaster. I will make sure that nobody goes on too long because we want to leave time for questions. This map is global, but it's far from complete. So if you come from a country with a different experience, it'd be great to hear your perspectives on it. We've got microphones. And so without further ado, let me get Antonio up here. Antonio, please.
Thanks, Carl. everybody. That Radiolab piece is definitely the best piece of journalism on CRISPR so far, I think so. If you haven't heard it, you probably should listen to it. I don't know if we have slides. Okay. All right, so the international discussion, where am I from? This is a satellite image of Cambridge, Massachusetts. You can see the Charles River. Down to the right is Boston. And so that's the MIT campus, and I've drawn a kind of a red circle around all those buildings there. And this area has become sort of the largest and densest concentration of biotech companies, of venture capitalists, of academic labs working on genetics really in the world. So I work right in the middle of it. It's really exciting and super convenient for me because just the main 3 CRISPR companies that have been established to make the CRISPR drugs, they're all within sort of walking distance of where I work, right in that circle. So that's where I am. Let's see. Okay, and what do? I have this quote also on my Twitter feed, and it's by Sidney Brenner, a Nobel laureate, and basically what he's saying, that in the course of science, the techniques are more important than the discoveries or the ideas. So I can justify why I only write about technologies like CRISPR, and before that, RNA interference, DNA sequencing, DNA chips, embryonic stem cells. I only write about the technology, the methods, the techniques. And here you can see what happens when some technique like CRISPR really works well. This chart only goes to 2015. I haven't kept updating it. This is just the number of papers. So it's just this exponential explosion, exponential wave. It's transforming science, potentially medicine, society. And so the scope of the story of this method is just huge. And the news cycle is, you know, it's already been going on for 5 or 6 years, and it's just going to, and it's gotten bigger, and it could get bigger still. This is the other sensibility I bring to the reporting. The finger is me, right? And the scientist is the one with the hand in the cookie jar. They are crossing some kind of ethical barrier, and I'm there to tisk them. So these 2 concepts, the technology and the cookie jar, come together in stories like this one. We had this on our cover a couple years ago. This is about the whole question of germline engineering. I'm actually decided that I would spend the rest of the time talking about the patents. Jennifer Doudna came to this conference. She made very interesting remarks, especially the Q&A was really interesting. People asked her about this patent fight that pits her university against where I work. The Broad Institute of MIT and Harvard owns the patent that you see on the left. So in her remarks, Doudna said that, you know, she found it discouraging that journalists would even write about this so much. And that it was kind of, you know, embarrassing to have all this money being spent on lawyers, tens of $millions a year in this colossal patent fight. So I just wanted to talk a little bit about how it originated for those of you who don't know. This slide represents the situation sort of mid-2015. On the left, you can see Jennifer Doudna, who is here, and Emmanuel Charpentier winning the Breakthrough Prize that's given here in San Francisco. It's funded by Mark Zuckerberg and other billionaires, so she's winning for inventing the CRISPR technology. But then on the left of the slide, you have Feng Zhang of the Broad Institute, and he has the patent on this. So how did that happen? How come the academic accolades are going here, but the commercial control is going to somebody else? That is the root of the whole dispute. It wasn't always so. If you rewind to 2013, all these people were part of one company. This was kind of the typical biotech company that gets launched inside that red circle I showed you. Some local venture capitalists, they pour a ton of money behind one company. They get all the key scientists, George Church, Zhang and Doudna in this case, and a couple of others, and they create a kind of a dream team company, right, that's going to own this landscape. But as soon as the patent issues, this dream team breaks up, and Jennifer Doudna actually quits the company, and she leaves. And it was interesting, for a while there, they still had this, the lower picture was still the sort of, even after she left, this company had this as their main image. I guess they didn't want to hire a new photographer because you could see Jennifer Doudna's back of her head is still there like a ghost in the machine. This is the timeline. In the middle of 2012, as Carl showed in his video, Jennifer Doudna and the teams that she worked with, they published this report of using CRISPR in a test tube. So they've kind of abstracted this technology, or they've abstracted this system out of bacteria, and they've kind of made it more efficient, a little bit simpler, and they can cut DNA at specific points in bacteria. And then immediately everybody realizes, well, you know, will it work in human cells? Can we edit human cells? People are off to the races to try and make that work. And just about 5 months later, come the first reports, including from Feng Zheng at the Broad Institute of MIT and Harvard, that they have made CRISPR work in human cells. Then, in the middle of 2013, is the founding of this company, Editas, where they all come together. But then, a year later, comes the patent. The patent was filed and prosecuted in such a way that it moved very quickly through the patent office, and people didn't know that it was coming. They didn't have sort of advance notice. And when it hit, it was kind of a huge bombshell, although the media wasn't immediately aware of this bombshell. It took about a year for the facts to come out. It was because to win the patent, the Broad Institute had provided like thousands of pages of references, documents, affidavits, testimony, a lot of which turned out to be critical of Jennifer Doudna, in some cases sort of, you know, kind of personal criticisms, that she needed help from some other scientists to do what she had done. So that was what caused the breakup of the team, and what was the seed of this animosity that drives this dispute. And so how did Broad win the patent? Because actually the patent examiner said you can't patent this because of the prior publication of Doudna. It's obvious what you're doing. Now the Broad Institute has argued that it wasn't obvious, that this use of the system, the changing of the system so it worked in human cells was its own invention. But they did have this problem with the patent examiner. And what's really interesting is the way they got around it is they did this. They just said, okay, well, we invented it first. We're gonna get rid of all your objections. We're gonna get rid of all your objections by saying that we actually had done this unbeknownst to anybody in 2011. I have run out of time, so here is the actual oath by Feng Zheng saying, I declare that I actually invented this in 2011. Okay? So, sworn testimony inside the patent office. Here is one of the exhibits. It's someone's computation notebook. It's incredible in this high-tech thing. We're still running on paper, you know. For my patent disputes, I always use national brand notebooks. And I was hoping that this would become like a court case, like O.J. Simpson trial of CRISPR, and that there would be people called to testify and cross-examinations and, you know, people breaking down on the stand, but it didn't happen. Instead, the Patent Office actually decided in favor of the Broad Institute. They just said, these are not the same inventions. You're not going to get your big court case. And so the Broad invention stands. And I'm out of time. Thank you very much.
Hi, morning. I'm glad to be here to share some of my observations on the coverage on genome editing in China. I'm Xiaoxu Chen. I'm a science editor at the Intellectual. The Intellectual is very young, 2 years old. So, in fact, I began to get into this field in 2015. At that time, the gene editing was very popular, and the media coverage on genome editing in China is basically focused on the research itself. The media explained what is gene editing, how the CRISPR works. But in China, the level of the science education level, I think, is not so high, and the public were not interested in CRISPR. So here I want to share the two main topics that connect the public with gene editing. One is ethical discussions, and the other is Ngiago gDNA system. That's about a personal story happened in China. In 2015, MIT Technology Review published this article. The author is just Antonio. And in this article, Antonio raised a question that the scientists were just trying to edit human embryos. So what does it mean? The germline the changes in our embryos, in germs or in eggs, could be transmitted into the descendants. And what will it be if this happened? Also, there's some labs mentioned, some labs in China were mentioned in Antonio's article, and it stirred up this heated discussion, concern on this issue. So many Chinese media quoted this article. Later, 1 month later, the article and Antonio mentioned came up. Yes, this is the first paper that added human embryo in the world. Though the embryos were discarded and they were non-viable, it was controversial. And in China, there are also heated discussions on this issue. There are several comments and other news coverage on this issue. The Meta Science, yes, where I used to work, organized a series articles about this issue. You can see their comments here. And also there is online debate on this issue. There's a debate among 3 scientists in China. One, Yi Rao, he is a neuroscientist at Peking University. Also, he's one of the editors-in-chief of the Intellectual. He proposed that human germline editing does not only engineer individuals, but also engineer our society. And also, 2 young scientists, Wei Wensheng, he's a CRISPR scientist at Peking University, and he just said, I'm optimistic about the future of human germline editing, but but still he's optimistic. And Li Mingwang, he's a professor at Zhejiang University, and he said the issues of genome editing is more related to regulatory framework. And the Intellectual covered this debate. And also we started an online survey. Do you agree to apply gene editing technology to human germline? And more than 1,000 people voted. And you see 65% disagreed and 29% agreed. Yes, that's the attitude of the public. It's a little different from that situation among the scientists. You see, the 2 scientists have hopes to edit the human germline. Only one opposed. Also here, next I'd like to share a special case about the coverage on gene editing in China. It's a story about an unknown scientist who was born in Shijiazhuang. Yes, that's where Feng Zhang came from. You see, Feng Zhang is a big potato, yeah, in the genome editing. And also, CRISPR is so popular and so powerful that people expect a lot. If we can find another tool just as powerful as CRISPR, and in 2016, Nature Biotechnology published a paper that reported NGRGO could edit mammalian cells. You see that here is the responding author of this article. He was unknown before, and he just worked as a low-ranking university in China. But MBT, MBT is a high-profile journal. That's an amazing story, yeah? And we just follow it, and he became a celebrity in China. But later, it proved that the NGRGO did not work, and in August, the authors retracted this article. You see, in China, people, it's hard for people, it's hard for science journalists to attract people to notice the scientific advances in China. But these two topics, I think they helped attract more people. First, ethical issues is a common concern, and the unusual stories seldom happened. They both helped connect gene editing with general public. Thank you.
Hi. Great to be here, and I'll try and skip through this because I'm really keen, like Carl and all of us are, to get your input into the discussion as well. But it's a delight to be here and hear everyone's perspective, because what a hot topic this is right now. Do we agree for our work? So welcome to Australia. Or is this Australia? Or perhaps that's Australia. And I can thankfully say that none of these images appear in the reporting of gene editing in Australia from my colleagues in the science journalism and other media. My own interest in reporting on gene editing centres around an interest I have in the intersection between science and culture and history and human rights and disability. So I got an Australian research lab to host and teach me CRISPR hands-on over a number of days in the context of a radio series that I was making about the history of eugenics and the prospect of a new eugenics. So a controversial frame, but a very potent issue. But I'm, of course, very interested in the fundamentals of the science as well, as we all are. So CRISPR in Australia. Research-wise, a lot is going on. I won't go into the detail, but it is an exploding arena. Publications are on the increase. We find it much easier to fund biomedical and applied research in Australia rather than kind of foundational science. So there's lots going on in terms of using CRISPR to study the genetics of a range of diseases and disorders, immunology, epigenetics, and a whole lot more. And there's also work being done on whether CRISPR and gene drives could help re-engineer invasive species like cane toads, which of course is a big problem for a big island like ours. Editing the germline, unlike China, unlike the USA, and unlike the UK, no one in Australia yet has applied for a licence to conduct gene editing on a human embryo. Certainly the idea of designer babies captures the media's attention and the public's attention, but under our current laws, which are strict, that prospect would be illegal. You can do research on human embryos if they're left over from the IVF process, but you need a licence. Interestingly, the Australian regulators tell me, though, that it's not clear whether the current legislation would allow research into editing human embryos. So there's some ambiguities and uncertainties right there at the moment, which leads me on to regulation, because this, like in many of our countries, is really potent at the moment. So we are on the cusp, as America and Europe are, of working out how to regulate this new form of gene editing. At the moment, the legislation governing all gene technology is under review in Australia, but prompted by CRISPR and what's going on, as is the Food Standards Code, because of related reasons. And scientists, I would say, from my dipping in and out of conversations with them and reporting, is they're quietly very anxious about this, and so are the food and livestock industries, because of course they saw what happened with the public discussion around GM and GM labelling. They're concerned that the research potential of CRISPR is going to be shut down before it really is able to evolve if the Australian public get a whiff that this is about genetic modification. So interestingly, if you track the language, and you probably have, a lot of the scientists are using the same language and arguments as they did in the late 90s to justify research on GM more broadly. And I think there's lots to be thought about there historically. And in their submissions to the review of our Gene Technology Act, they all make the same argument, that the current Gene Technology Act is outdated and obsolete. Australian scientists are in fact pushing quite hard behind the scenes to have CRISPR and related editing techniques mostly excluded from regulation. Now, you'll find that in your own countries as well, and I think that's a very interesting reporting point for us. So they want CRISPR clearly delineated from traditional genetic engineering techniques like recombinant DNA. So, for example, if you're using CRISPR, they say, to delete a genetic sequence or insert a genetic sequence that is of the same species as the organism that you're manipulating, then this, the argument says, is no different from products of chemical or radiation mutagenesis or traditional breeding techniques, and none of those are currently regulated. It's just a faster way of doing what comes naturally, basically. And they say unless you're inserting a gene from another species to create a transgenic organism, then CRISPR products should not be considered GMOs, and so they shouldn't be regulated. Isn't that an interesting argument right now? This is even though the off-target effects of CRISPR are still quite uncertain. So there's an area of reportage that we should pay attention to. And just yesterday, the regulator put out an interim notice saying it's likely that what they'll do is that they'll, if a scientist is inserting anything as big as a gene or bigger, then that will probably be regulated as a GMO. And I can imagine right now that scientists are in a big dither about that. So that's an interesting development. So we have a role to look beyond the semantics, I think, of this, because it's quite seductive, the arguments that get made, but we need to interrogate the detail of the science closely, report on it, report on what we do and don't know so far, and really promote those conversations. Partly because there's also, in all of our countries, I would wager, those technical advisory bodies that are informing regulation, there are arguments and claims that they have major conflicts of interest. And even the scientists on those committees acknowledge that conflict of interest. You want genetic experts on these committees advising regulation. Absolutely you do. They know what they're doing. They know the science. On the other hand, they have financial and scientific and research interests in the science that they're advising on and what regulation should happen. And so that's a really, you know, those governance issues are quite substantial, they're delicate, and that's where we can have a role too. Are those committees made up of a range of voices? You know, this is not just a technical issue, this is a profound social issue. Is feeding the world as much of a political problem as it is a social problem? What about the tensions between community needs and the needs of agribusiness? And, you know, if we use gene editing to remove the horns of dairy cattle, is that the best way to respond to the animal welfare issues of the dairy industry and mass livestock production? Don't know. So, to wrap, one particular area that I'm interested in, and I wanted to touch on this really briefly because I'd love to hear your thoughts and the thoughts of the panel on this, is disability and identity. So last year I made this radio series on the history of eugenics, and I did CRISPR as part of that, or partially. It's kind of a mocked version of CRISPR, and recorded that. And I spoke to an elderly survivor of America's mass sterilisation laws. Her name was Rose, and her story almost precisely mirrored that of a young woman called Carrie Buck. And just nod your head if you know the story of Carrie Buck. So she was a young woman in the early 1900s. I'll just whip through this. She was taken out of school. She was raped by the nephew of her foster parents. She had a child. The child was taken away from her. She was branded an imbecile by her foster parents. She was incarcerated in a colony with lots of other people who happened to be there because they were either disabled, had a child out of wedlock, or for whatever reason. Often they were just poor. And she then became the subject of a Supreme Court case that ultimately had her forcibly sterilised because she was genetically inferior, and that led to multiple eugenic sterilisation laws across America. Up to 70,000 people were sterilised. Not that long ago, in fact. It's astounding. And when you dig into the science of that whole case, and I'll just whip through this, you know, and you interrogate the way in which doctors and scientists and people with power behaved and drew on the fledgling genetic science of the time, it's a really sobering lesson in how new science can be misappropriated and misused and abused. And recently I gave a graduation speech at a university in Australia where they were all graduating from bioscience and biomedicine, and when I did my preparation for that and I looked through their whole curriculum, I went through it with a fine-tooth comb, there was nothing that those students had studied about the history, sociology, ethics, politics, nothing on any of that to do with genetics, and they are graduating from a top university as biotechnologists. So I think as reporters, I hope that we consider the voices of those who are facing the prospects of their own eradication, people with the lived experience of disability, who are very vibrant and vocal now. They are the carry bucks of the present. We are also in a time where we're saying we're pathologising poverty again. And so I think we have a duty of care about what range of voices we bring into our stories about new genetic technologies. Thank you. Look forward to the discussion.
Hello, everybody. I have two questions for you. The first one is, how many of you are from Latin America? So the second question is for the people who are not from Latin America, and who has here a CRISPR news story from Latin America? Do you? Did you? One? Or two? Okay. Well, there is some work going on in Latin America. Romina Bebacqua, who was 30 years old, and her group, and her team eliminated the gene responsible for mad cow disease using CRISPR in 2016, founded by the government of Argentina. And they use cow cells and cow embryos, and that potentially could be used to develop disease-resistant livestock. And there are more researchers using CRISPR in Argentina. There are 8 groups with these goals: to produce animals to help people with diabetes, lactose intolerance, and cardiovascular diseases, and to increase the production of crops. What they can do under the current laws in human, okay, of course, they can work on germinal line, on human germinal line, sorry. With plants and animals, we have a resolution, a law. It's the only, I think it's the only one in the world. If the CRISPR organism doesn't have any new genetic material in a stable manner, it isn't labeled as GMO. Yes? It will, and well, we are journalists, and we have to think how we work. Since 2013, more 400 news stories about CRISPR were published in Argentina by different media, print and digital media. And if you compare the number with the New York Times. I found 60 news stories by the New York Times. And most of the stories in Argentina are about CRISPR in USA and China. They focus only on the perfect baby. They don't provide a framework with ethical issues, but they have not hyped CRISPR stories as much as the stem cells stories were. And they didn't include church comments inside science news stories as much as they did when the reproductive issues were in the news in the past. So I think we are living a good moment for genetic issues, and we have to take the opportunity. Some tips cover what Latin Americans are doing with gene editing. Give voice to women in science in your countries, on your stories. Look for news sources for several fields, like biotechnology, law, bioethics, psychology. Find out if there are public conversations about CRISPR and other techniques, and put the religious source in perspective. So these are my opinion. I would like to thank the organizers. I would like to Carl. I would like to two friends of mine, two US friends, Infe and Bill, who helped me to write better this presentation. Thank you so much.
Thank you very much. I often joke that I've been a science reporter for so long that I covered the extinction of the dinosaurs. It's not been quite that long, but I have seen a thing or two in my time. And so, if I may, I'd like to offer a historical perspective as well as talk a little bit about how CRISPR has been reported in the UK. So, just on that first, this is from the website of Britain's best-selling newspaper, The Sun. Gene Genie, embryos edited to remove killer mutations, like the one that nearly killed Fabrice Muamba. And in big capital letters, it could save millions. Fabrice Muamba was a Premiership soccer player that collapsed during a live television game. And he survived, despite the fact his heart stopped for 78 minutes. And the reason I mention it is that the best-selling news, this was front page news, this was the lead story about this particular piece of research. And it was written in a way that made sense to the average person. And a month later, the same newspaper reported on the knocking out of the OCT4 gene by a team at the Francis Crick Institute. It says scientists could say it could signal the end of inherited diseases. So not so long ago, The Sun might have been one of those newspapers that would have talked about designer babies and brought in objections from pro-life groups, but not anymore. The messages, not just from The Sun, but other British newspapers and broadcasters, are on message. Now, this may be due in part to the greater organisation of university and industry press officers, coordinated by the British Science Media Centre, which is an organisation funded by research organisation and industry groups to try and get their view of science across. And it seems like they've succeeded. Now, having The Sun on your side is absolutely huge. The Murdoch-owned paper often claimed that it could decide the outcome of elections. So to get it on side on controversial research marks a sea change in the way in which science is reported. And so, if I can reel you back when I was starting out as a network reporter, these were some of the headlines that we were seeing about concerns on the MMR triple jab vaccine, which you're having in the US right now. and concerns over the safety of GM crops. And that alarm on MMR meant that lots of children were not vaccinated and had measles. And GM crops, to this day, are still banned from supermarket shelves. So it can't be right, but scientific voices were drowned out. But as the scientific community began to organise, as the Science Media Centre got into gear, this began to change. Now look at this from another tabloid, the Daily Mail. It says that the benefits, so the legislation was changed in order to allow British researchers to carry out experiments on human embryonic material, stem cells. And the Mail says, which was on the forefront of the anti-MMR and anti-GM campaign, says the benefits of stem cell research outweigh ethical concerns. Researchers may think it's possible to cure Alzheimer's disease and grow spare part organs. And a year later, in 2001, the Mail was saying gene therapy could cure cancer. Now, of course, this is from the Cancer Research UK. It talks about, 16 years later, gene therapy is just a promising area of research. But back then, there were high hopes for gene therapy. It was hoped that the Mail again, the Mail on Sunday, scientists are poised, I just jumped ahead, could we be on the verge of the greatest medical advance ever seen? Greater than the defeat of smallpox or cholera, the defeat of time itself. Lovely writing, but I think, the answer to that is no. And again, there were concerns by the biotech industry that synthetic biology should not go down the route of GM crops. And so the Telegraph reports scientists are poised to create the world's first synthetic microbe that could lead to an endless supply of biofuel. So I think you know where I'm going with this. What's happening to our coverage of gene therapy. When did we think that our job as science and health writers was to do the PR of scientific organisations? Our job should be to challenge and question, whether gene editing will lead to the end of inherited diseases, rather than report blankly that it's just going to happen. So that's what the World Federation of Science Journalists, one of the people that helped organise this conference, was created for. And that's what we're trying to do with the ABSW, the Association of British Science Writers. We're trying to develop a culture where we up our game, that we don't fall for obviously the people are enthusiastic about technologies, but we need to do better. We need to up our game, because obviously it's great that we're not, reporting the fact that MMR causes Crohn's disease or GM crops harm your health. But by reporting exactly what the scientific community wants to report without questioning, are we not leading to another kind of misinformation? And so finally, I'll just leave you with this thought. I just wonder, in 20 years' time, at the 20th World Conference of Science Journalists, whether we're going to be talking about, we're all going to be here, rejuvenated, sorry, this is our, rejuvenated, and talking about the end of all inherited diseases, or even one. I suspect not. Anyway, thank you very much. I avoided Carl's notebook, so that's good, leaving you time to talk.
Okay, so we have a microphone there and a microphone there. So please, if you have a question, please get up there. And while you're doing that, I will cannibalize some of Paolo's extra time to ask Antonio a little bit more about patents. Because there's been some, this is an ongoing story, and just in the past few days, there have been more development. So where does the patent fight stand right now, and what's your prediction about how it's going to shake out?
Let's see. It's complex enough that I actually don't know I actually don't know the answer, but currently the Broad has this patent that for the time being, is going to stand. Berkeley has an appeal that's going to a district court that handles patent cases. So they're going to try and reverse the decision by the Patent Office, which upheld the Broad patent. And their own patent may at some point issue, and so there could be these two patents at the same time. And then in other countries, I think in the UK and maybe in China, the Berkeley patent has actually issued. So then...
So Berkeley has the patent in China?
I think so, yeah.
Can you pass from the Cherchier one? So Berkeley's in charge in China?
What I'm sure is that Broad Institute has no patent in China, but the patents on CRISPR has been issued in China. Perhaps it's owned by Berkeley. Okay. Yeah.
I'm sorry, Antonio, but I just wanted to clarify.
Each country could have a different situation.
So this will be just a fight that's going to just play out across the world for a while.
It is, and it'll change too, because as people develop sort of follow-on technologies, there'll be other patents and other important things that happen. So, you know, it's not really clear how important this patent fight will be. It's sort of a simplification of the whole issue, but it's interesting.
So someone goes out and finds a new strain of bacteria that does an even better kind of gene editing, hit that patent, and this is all old news. Okay. Yes, question please.
So, piggybacking on that, should these techniques be patentable at all?
Who wants to take that? Do people have the right to patent CRISPR?
Just in the way, like people have discussed, like we don't have this. You might have debated before, you know, the ethics of making embryos and stuff. And we've kind of progressed beyond that. We don't worry about those sort of religiously motivated things as much anymore. Similarly with the patents, I mean, it's widely accepted that patents are kind of a good thing. And so the voices questioning whether they should exist at all are few. I can't answer the legal question. I think there is some question, you know, if something exists in nature, you can't patent it. What they've done here is they've taken something from nature and they've tweaked it, so I don't think it's going to get knocked out by that objection. I think under patent law, it's definitely patentable. Should it be patented? I think the question in this debate is, should it be licensed exclusively, which is what these universities have done. They've licensed exclusively to companies that their faculty started and have millions of dollars of equity in, and that they have equity in. So I think that's the bigger question.
What's your sort of sense of the, I don't know, the kind of the personal politics of the whole patent battle? You know, I've had people come up to me and go, well, there's sexism in action right there with this whole patent thing. You know, that the women aren't getting the credit and the men are, and, you know, so there's been a whole range of interesting interpretations, and I think as science reporters we can kind of extend the patent story out beyond the business domain, and I'm interested in people's thoughts about that, particularly you as following it all so closely.
Can I dodge that question? We'll take one from the floor. I don't want to
sure.
make a pronouncement.
Time is of the essence. Anybody want to take that one? All right. Okay.
Hi, Lisa Krieger at the San Jose Mercury News. For those of us who are trying to keep up with the science, this is a reporting question. How do you learn what's in the pipeline of the various labs before we're sort of, you know, ambushed with a paper that's published?
Yeah.
This is for Carl and Antonio or anyone else.
Yeah, I mean, I'd be curious what other people have to say, because there was a time where you could just sort of, you could keep up with it, and I mean, and you're like, oh, okay, and then there's a gene drive thing, and there's a gene therapy thing, okay, that's fine, but then it just sort of coned out, and like Antonio showed, it's just, it's too much, and then nestled in all that stuff are the really big things that hit.
Right.
So I personally just keep an eagle eye on the journals coming out. I just talk to people in the field and just be like, what do you think's going on? And you know, preprints are starting to become a factor. So bioarchive, you have to keep your eye on bioarchive. But if you're not already, go to bioarchive, because people are just throwing their stuff out there, and then they become subjects of debates. So it's hard. It's hard. I can't. I don't know. what are people's secrets for keeping up with CRISPR?
I'll just quickly add that I hadn't done a heap of reporting on all the developments in CRISPR at all. But so what I did is I rang one key player in the field who is involved in trying to network scientists in Australia who are driving CRISPR, and I spent about 3 hours on the phone with him, and he just talked me through everything that everyone's doing. So if you can find one key kind of network of scientists, I would really recommend having that conversation. It's extremely helpful.
Although, there's so much vying for patents and for prizes and all that. Like, I'm a little suspicious sometimes. I'm like, so what do you think of what they're doing? You know, they'll be like, oh, that's garbage. Don't listen to them.
Yeah.
I mean, it's you got to be careful not to rely on one person. Oh, Australia. Everyone's nice in Australia, right? Okay. Anybody else want to respond to that one?
Antonio, yeah, how do you keep up?
What's your secret, Antonio?
Well, I have an entire spider's web that covers all of CRISPR, so I feel it jiggling, and then I try to get to the news. I mean, I have the luxury of spending most of my time on this technology, so...
So CRISPR is your beat now, basically.
Yeah, I write about other things, but mostly that. In fact, like you, I'm kind of, it's taken over my life. So we could talk about it afterwards. I mean, if you're spending all your time on something, then, eventually you could know things.
You know things. There are
Thank
things.
you.
Yes, sir.
Okay. Yeah, so actually Natasha's picture just now used is quite similar to what Greenpeace used against the GMO. So GMO could be one of the most controversial and most widely lasted topics. So right now we come to the, also the science journalists get mostly involved in GMO issues. So now we come to the genomic editing. Do you think we as science journalists should get a similar level of involvement in the genomic, in the gene editing debate, the gene editing report, just like what we have been done in GMO issue? Thank you. Of you.
Yeah, Paula.
Natasha mentioned, a few of the speakers mentioned that there's a move to try and make gene editing not in the same category as genetic modification. Exactly the same thing's happening in Europe. The European Court of Justice is making a ruling next year as to whether something that uses gene editing that doesn't introduce a modification should be counted as not gene editing. I think that certainly in Europe, where GM crops are banned, there's a real concerted effort to rerun the GM debate but win this time. I think that's in their dreams. I think, as it emerges, people realize that they're being conned. I mean, the issue should be about whether these things are safe or not, rather than trying to win some sort of legal battle. So I think that's going to be a very fruitful area of reporting, certainly in Europe.
I wonder, though, because I think a big part of the reaction to GMOs, at least in the United States, I'm not sure how it was in other parts of the world, but was a feeling of foreignness, that there's something unnatural about, taking a gene from one species and putting it into another. It was almost like some bizarre bestiality kind of reaction that people would have and be like, this is unnatural, this is against God's will almost. And whereas with CRISPR, it's just like, oh, we're just making, we're just tightening the knob a little here and there, and the sort of the foreignness is not a factor. So I'm not convinced that there's going to be a CRISPR backlash, just because of that sort of motivation.
So that's the argument that biotech companies are making, and that's the argument that's...
Closer, mic closer.
Sorry, that's the argument that the biotech companies are making in Europe, that it is natural just because it involves an intermediate genetic modification stage. It is just natural. Frankly, I don't know. We'll see who's right. When we're at the 20th World Conference, rejuvenated as to whether, which way it goes. But my instincts are that in Europe, the public will see this as a bit of a con.
Anybody else?
Yeah, the GMO, as the anti-GMO movement, yes, in China, the Ministry of Agriculture hasn't issued as much food in China, but I think though the media is deeply involved in reporting GMO, it seems that it has no effect on the decision of the government and for the information of the public. But referring to the gene editing, I think we have to learn lessons from the GMO coverage, but balancing and making the public participating, also the scientists participating in discussion. Because in China, there are some celebrities, they led the discussion, but the scientists' voices has not been widely heard. Yes, that's it.
Just before I take the next question, I want to see a show of hands. Paula was talking about what's going to be happening in 20 years. So in 20 years, do you think that there will be a human being walking around who was the product of a CRISPR-modified embryo?
I think the techniques...
Yes? Could you take the microphone? Oh, sorry.
And interesting to hear Jennifer Doudna imply the same when she was speaking the other day, and I'm inclined to believe her.
Valeria, what do you think?
I think CRISPR is going to be used in somatic line for.
But what about germline? Human germline?
I don't think so.
You don't think so?
No.
Yes. Next question.
Yeah. My question is split into two parts. One is specifically for Valeria on this use of CRISPR technology, or rather the gene drives for mosquito control. What has been the experience in Latin America in the sense that has the media debated over the implications of the gene drive technology, or have they just restricted themselves to designer babies and not written about the implications, ecological implications of gene drive? And my second question is addressed to everybody else also. Have you as journalists been accused, or rather not maybe directly accused, but a very subtle hint that you media are going to kill this baby or this technology even before it is born with your questions on ethics and your questions on appropriateness, etc.?
All right, two questions. So the first question to Valeria. Gene drive is being explored as a possibility for controlling diseases like Zika or malaria by basically driving a resistance gene into a population, or maybe even eradicating a species like mosquitoes. So what's going on with that?
Well,
in Latin America?
the use of CRISPR for gene drivers is not a hot topic, at least in Argentina today. But I think maybe we should think more about it. But the government in Argentina organized a conversation with the INSER of France, the institution of France last year, and they were different researchers from Latin America were discussing the different uses. They didn't reach any declaration, but they were discussing these kind of uses.
And the other question, whoever wants to take it, I mean, do you think scientists are looking at us journalists as, you know, suffocating this baby in the crib?
As I was saying in my talk, I think the opposite's true. In the UK, the coverage has not only been uncritical, it's kind of praised the kind of future. We're going to eliminate all inherited diseases. There hasn't been, as in the coverage that I've read, a single dissenting voice. I mean, the word designer babies might be mentioned, but disability groups are not represented in this debate. Scientific question marks as to whether it really is possible have not been raised. So I think the opposite is true in the UK.
I think we're at a very interesting time in terms of our own reportage on this whole issue. My conversations with scientists have suggested that they are very anxious about what's to come, but that hasn't precluded them from being very open about some of those concerns, but also just open about their own wrestling with the ethical questions. My concern is, though, like Jennifer, I don't know who saw Jennifer's talk this week, but Jennifer Doudna, she said this is very important, lip service, and she's been doing a lot of that. This is very important to consider the ethical considerations. I have concerns, I'm anxious, etc. I had a nightmare about Nazis, etc. But they need to be, I believe personally, they don't have the skills to work through the ethics of this stuff. They need to be coupling up with social scientists and others to think about frameworks to have these conversations. They do not have the skills to do that themselves while they are doing the science. They need to be working in companionship.
Can I just quickly?
Yes, please.
I think that there's many things that are that people don't want to have known, and that they're lurking and sometimes in plain sight. In Jennifer Doudna's dream, the famous dream where someone, a colleague says, "Come meet my friend Hitler." In the many recountings of the story, we never hear who the colleague is. So there's a secret in plain sight. Does she know who she thinks in her dream is friends with Hitler? I would like to know. I didn't ask this question.
You got to get your spider network on that one. Okay.
Just sharing a piece of information. Apparently, in India, the definition of GMO, because now it's coming out, the whole CRISPR debate. For some reason, somebody decided to include deletion or insertion of genes as a part of genetic modification. So inadvertently, not by design maybe, but by default, this piece is already being covered by regulation.
That could have a big impact, yeah.
Yeah.
I'm afraid we have time for one more question, so sir, you can round it out.
Okay, so we've heard a lot in the media about the famous battle over the patent, and I'm wondering if you can give us a little bit better idea of really what the stakes are, what such a patent means. If it turns out to be Broad that comes out on top, what kinds of things could they claim authorship of, and how does it all work exactly?
Antonio?
I mean, the patent has already had, the existence of the patent allowed the company to raise money, this Editas, and the other companies have raised money on the promise of their intellectual property portfolios as well. So you can actually raise money, go public, sell shares to the public, collect that money, cash out, become a millionaire while your product is still undefined. So the concrete value of the product, the patent, is the ability to cash in on CRISPR long before you've actually done the work. The answer to your question is so long and complex, but I just wanted to leave you with that one thought, like that's actually how it was used in practice so far.
Great. Okay, well, thank you so much for coming, and please have a hand for our panelists.